cellasys 8 is a microphysiometric assay for monitoring living cellular models. The assay itself runs for 24 hours, and you can get results within 2 days. The assay measures the rate of extracellular acidification and the electrical cell substrate impedance in real time and without a marker. The cellasys 8 assay allows positive, negative, and blank controls. It comes with 4 replicates of the actual test. Here it switches between the reference medium and the test medium. The unique feature of this assay is that within minutes you can see the response of the cellular model towards the test medium. Recovery effects and effects of repeated doses are recorded too. The cellasys 8 assay is performed with the 6-channel emolla IVD bundle. The setup consists of a cell culture incubator for temperature control, 6 emolla devices, a peristaltic pump, 6 fluidic modules, and a laptop for control and data acquisition. Accessories required are 6 biochips with O-rings, 6 reference electrodes with barrels, 6 fluidic heads, 18 100 milliliter GL45 flasks with distribution caps and Lure steel needles, 6 flasks for sterilization and cleaning solutions. Finally, you need the following common consumables and reagents: double distilled and autoclaved water, 70% ethanol solution, 2.5% sodium hypochlorite solution, sterile pipettes, sterile tweezers, 1 large Petri dish, and of course you need your cell culture medium and the living cells. To start the preparation process of the assay, you have to activate the power supply. Start the main power supply, the computer, and the 6 emolla devices. Click in the pump cartridges. Prepare the reference electrodes with the barrels. To do so, screw the reference electrodes into the barrels and connect them to the fluidic system. To sterilize the whole system, add about 50 milliliters of 2.5% sodium hypochlorite solution to each of the 6 flasks and insert the 4 Lure steel connectors. If the software app Dahlia client has not been started yet, start the app on your computer. Choose the sterilization application and press Start. The sterilization process runs automatically for approximately 25 minutes. The next step is to prepare the reference medium, the test medium, and the medium for the positive control. Fill 7 flasks with 40 milliliters and 1 flask with 25 milliliters of the reference medium. And add 500 microliters of 10% SDS solution to the 25 milliliter flask. Then close the flasks with the distribution caps and connect the Lure steel needles. After that, use the test medium to prepare 5 flasks of 40 milliliters and 5 flasks of 25 milliliters. Repeat the known procedure. Add 500 microliters of the 10% SDS solution to the 25 milliliter flasks and connect the distribution caps and Lure steel needles. The prepared flasks must now be connected to the 6-channel emolla fluidic system. For emolla 1, place the 25 milliliter reference medium flask in position M2. For position M3 and M4 of emolla 1, use 2 40 milliliter reference media flasks. The setup is the same with emolla 2 to 6. 1 flask with 25 milliliters test medium needs to be put to M2, 40 milliliters test medium should be placed to M3, and the 40 milliliters reference medium to M4. Please make sure that no tubes are compressed or otherwise blocked. In order to pre-fill the fluidic system with the reference medium, you need to run the pre-filling application again. If the previous application is still running, press Stop. you have to load the pre-filling application and press Start. The script will run automatically for approximately 25 minutes. After this process, the 6-channel emolla is ready for the assay. Now all you have to do is turn on the incubator. The medium in the system will be tempered to 37 degrees Celsius overnight. To prepare the 6 biochips, let them sit with 70% of ethanol for 20 minutes in the laminar flow. Afterwards, remove the alcohol by washing the biochips twice with double distilled water. To seed the biochips, use 300 microliters of reference medium with a desired number of cells and fill it into 5 of the biochips. 1 biochip without any cells in the reference medium should be used as a control. The preparation of the assay is now completed. Place the biochips in the CO2 incubator overnight so that the cells adhere to the substrate. Since the heating process of the medium might have created some air bubbles overnight, you should start the next day with another pre-filling sequence. Use the application and press Stop and Start to perform the pre-filling sequence. It will run again for approximately 25 minutes. Don't forget to press Stop afterwards. Now prepare the O-rings in 70% ethanol and the fluidic heads in 2.5% sodium hypochlorite in the laminar flow. Let them sit for 20 minutes. Afterwards, rinse both items using double distilled water. Take the biochips and examine them under the microscope. Ideally, the biochip should be about 80% confluent, except for 1 biochip, the one with no cells, which we use as a control. Now, in the laminar flow, place the O-rings into the wells of the biochips. Please make sure to start with a biochip without any cells. Press the fluidic head completely onto the biochip and close the lure connectors and the clamp. To install the biochips in the emolla systems, open the clamp, place the biochips into the socket and close the clamp. Then, open the lure connector of the biochip and the one in front of the reference electrode and connect the biochip to the fluidic system. Please note that the biochip without any cells must not be inserted into emolla 1. Usually, it's placed into emolla 2. Now, everything is ready to start the cellasys 8 assay. To do so, you have to load the cellasys 8 application and press Start. The cellasys 8 protocol will now run automatically for the next 24 hours. After 24 hours or later, press Stop to terminate the cellasys 8 application. Now the collected data can be exported via the Database button in the menu of the Dahlia client software. The data can be exported to a simple CSV file or an HTML-based EXP file, which contains every detail of the experiment. To prepare the next cellasys 8 process, or to shut down the 6-channel emolla, you need to do the following. Disassemble the biochips with reference electrodes. Clean them with double distilled water. Store the barrels in 3 molar potassium chloride solution. Prepare 6 flasks with double distilled water. Start the cleaning application sequence to flush the system with double distilled water. After 25 minutes, release the pump cartridges and shut down the system. Now it's all done.